I want you to take a second and think about a highly stressful situation you've faced recently.
Mm,
Maybe it was a profoundly toxic workplace that you just had to endure day in and day out. Or perhaps, an incredibly difficult, draining personal relationship.
right, the kind that just exhausts you.
Exactly. When you were in the thick of that stress, how did you actually react? Because if you look closely at human behavior, we tend to have two completely divergent responses to severe adversity.
I really do.
Yeah. Sometimes your default response is to just grit your teeth, put your head down, endure the misery, and simply wait for things to eventually improve.
You absorb below.
Right, you absorb it. Yet, in other situations, under seemingly similar amounts of stress, you might feel this sudden, intense, almost undeniable drive to completely change your environment,
Yeah, to just quit the job.
to leave the relationship, to radically alter the trajectory of your life. Why do we have these two completely different modes of handling adversity?
It's it it's a profound question. And the answer to why you respond in these two drastically different ways actually lies deep within the microscopic architecture of your brain.
Okay.
Specifically, it comes down to the neuroscience of a single, highly sophisticated neuromodulator,
Yeah.
serotonin.
Serotonin.
Yeah.
Yeah.
Now, for a very long time, pop culture has completely misrepresented serotonin.
Yeah.
It's constantly referred to as the simple happy chemical,
Oh, absolutely. You see it on T-shirts. You hear it in casual conversation.
sold to us in marketing copy, for supplements, all of that.
Right.
But the reality is far more intricate and honestly far more fascinating. Serotonin is not a joy delivery system.
It's not.
No. It is a complex, masterful regulator that dictates exactly how you as a human being cope with adversity and stress.
Okay, let's unpack this because the stack of authoritative neuroscience journals we have to go through today is, it's staggering.
It's quite a stack.
It is. Our mission for this deep dive is to completely dismantle that happy chemical myth. We are going to explore the cutting-edge neuroscience of passive and active coping. Mm-hmm.
And how these two distinct survival strategies are mediated by completely different serotonin receptors.
Mm-hmm.
And to make sure we're giving you the most accurate, applicable information possible, we are going to focus exclusively on human studies and behavioral interventions.
Which is crucial. No mouse models today.
No mouse models. We're looking at literal structural changes, vast network connectivity across the human brain, and the specific regional activations that govern your daily decision-making under stress.
The human brain is just a marvel of biological engineering. And to truly understand human behavior, we have to connect these microscopic receptor functions.
Like molecules shifting across a synapse.
Exactly. We have to connect that to the bigger picture of how you navigate your life. We'll be looking at how a chemical produced in a tiny cluster of cells at the base of your brain manages to control everything from your impulsivity to your capacity for patience.
Your sensitivity to losing money. Yes. And your ability to inhibit a bad habit. It's a deeply analytical journey, but by the end of it, you'll have a foundational understanding of the biological machinery that drives your reactions to the world around you.
I am so ready for this. Let's start by laying the foundation. There's a groundbreaking framework that really sets the stage for everything else we'll discuss today.
The 2017 paper.
Yes, coming from a highly influential paper published in 2017 by Carhart Harris and Nutt in the Journal of Psychopharmacology. It's titled Serotonin and Brain Function, A Tale of Two Receptors.
A classic at this point.
It really is. This paper completely shifted how we think about coping mechanisms. But before we get into the two receptors part of the tale, we need to understand the physical real estate we're dealing with.
Right. Where does the serotonin even come from?
Exactly.
To map that out, we rely heavily on a comprehensive 2010 paper by Yves Charnay and Lucien Leger, published in Dialogues in Clinical Neuroscience.
Brain serotonergic circuitries, right.
That's the one. In this paper, they outline the structural anatomy of the human serotonergic system with incredible precision. What's staggering is that out of the roughly 86 billion neurons in your brain.
Mainish 86 billion.
Yeah, only about 350,000 of them actually produce serotonin.
Wait, 350,000 out of 86 billion? That is a remarkably tiny fraction for a chemical that supposedly regulates our entire mood and survival strategy.
It is a microscopic fraction.
Yeah.
They're clustered in a tiny primitive region of the brainstem called the Raphe nuclei, with the vast majority in the dorsal and median rock nuclei.
Okay.
But from that tiny centralized location, these neurons send out incredibly long, extensively branched axons that project upward and outward. They innervate almost every single region of the human brain.
It's like a massive root system growing out of a tiny seed in the brainstem,
Right.
reaching all the way up into the highest cognitive center.
Precisely. They project into the amygdala, the hippocampus, and all the way to the prefrontal cortex.
But here's the thing I always found weird about serotonin neurons. They aren't wired like typical neurons, right?
That's a crucial distinction. Many of these axons are non-myelinated.
Meaning they lack that fatty insulating sheath.
Exactly, the sheath that allows for rapid point-to-point electrical signaling. So a lot of serotonin transmission happens through what we call volume transmission.
Volume transmission.
Yeah. Instead of just firing across a tiny closed synapse to a single neighboring cell, these axons have small vertices, little bulges,
Right.
and they release serotonin into the general extracellular space. It essentially bathes entire networks of neurons at once.
So it's less like a text message to a specific person and more like a radio broadcast.
That's a perfect analogy.
Yeah.
And how those local networks respond to this serotonin broadcast depends entirely on which specific serotonin receptors are present on their cell membranes.
Which brings us back to that Carhart-Harris and Nutt paper. They argue that human behavior under stress is largely governed by a bipartite model, a two-part system.
Right.
And it's mediated predominantly by two specific diametrically opposed receptors, the 5-HT1A receptor and the 5-HT2A receptor.
Yeah.
Let's look at the first half of that model, passive coping. The paper defines passive coping not as giving up, but as tolerating a source of stress without necessarily changing the situation itself.
It's the ability to endure.
Exactly. When you're in that toxic job and you just put your head down and do the work without quitting, you are engaging in passive coping. And this is mediated by that first receptor, the 5-HT1A receptor.
If we connect this to the broader neurobiology, the 5-HT1A receptor is fundamentally inhibitory.
It calms things down.
Right. It's largely located postsynaptically in limbic regions and the prefrontal cortex. When serotonin binds to the 5-HT1A receptor, it hyperpolarizes the neuron.
Making it much less likely to fire an electrical signal.
Exactly. It acts as a biological brake. When you're exposed to stress, your limbic system naturally becomes hyperactive. The 5-HT1A receptor engages to quiet those hyperactive stress circuits.
So it promotes emotional blunting, patience, endurance.
It reduces anxiety so that you can simply survive the adverse conditions. And crucially, this is the exact pathway that is largely enhanced by conventional SSRI antidepressants.
Let me pause you there. If the 1A receptor is hitting the brakes and calming the brain down, how does that actually help someone in a toxic job?
Well,
Doesn't that just make them numb to the abuse? Isn't emotional blunting a negative side effect?
that's a common perspective, especially when we talk about the side effects of SSRIs. But from an evolutionary standpoint, emotional blunting is a highly effective survival strategy.
Really?
Yes. Think about it. If you are in an environment where the stressor is unavoidable, say a brutal winter thousands of years ago, or a modern economic situation where you absolutely cannot afford to lose your paycheck.
You're trapped.
Right. Panicking every single day will deplete your physiological resources. You will burn out, or you will make a rash, dangerous decision.
So the 1A receptor's inhibitory effect allows you to detach just enough to keep functioning.
Exactly. It lowers the metabolic cost of the stress. It's not about thriving. It's about surviving until the environment naturally improves.
Okay, that makes a lot of sense. So it's essentially a shock absorber. It doesn't fix the bumpy road, but it stops the car from shaking apart.
I love that analogy.
But then we have the exact opposite side of the coin, active coping.
The 2A receptor.
Yes. Carhart-Harris and Nutt define this as actively dealing with psychological pain by changing your relationship to it or changing the environment itself, quitting the job, having the difficult conversation.
And this drive is governed by the 5-HT2A receptor.
Right.
What's fascinating here is that the 5-HT2A receptor is in diametric opposition to the 1A receptor.
Yeah.
It is excitatory.
Excitatory.
Yes. When serotonin binds to it, it depolarizes the cell, making it more sensitive and more likely to fire. Some authors argue that activation of the 5-HT2A receptor opens up a biological window of plasticity.
Window of plasticity, meaning the brain is ready to change.
Exactly. Under normal, everyday conditions, the inhibitory 1A receptor dominates, keeping you calm and stable. But they propose that during states of extreme adversity, when the stress reaches a critical, life-threatening threshold, serotonin release floods the system so heavily that it begins to aggressively engage these excitatory 2A receptors.
And that triggers massive neuroplasticity.
Yes, essentially forcing the human to learn, adapt, and change their environment to survive.
Going back to that driving analogy, if the 5-HT1A receptor is your car's shock absorber, the 5-HT2A receptor is your steering wheel.
That's a great way to visualize it.
When the road gets so impossibly dangerous that the shock absorbers are no longer enough to save you, the 5-HT2A receptor allows you to turn the wheel and navigate completely off that bumpy road to find a newly paved highway. It provides the plasticity to change your path.
And what makes this cellular architecture truly elegant is where these receptors are located. In the prefrontal cortex, up to 80% of the main computing cells, the pyramidal neurons, co-express both the 5-HT1A and the 5-HT2A receptors on their very same cell membrane.
Wait, on the exact same neuron?
On the exact same neuron. You have these two diametrically opposed receptors engaged in a constant, delicate tug-of-war.
Wow.
The 1A receptor pulls toward endurance, inhibition, and passive tolerance. The 2A receptor pulls toward excitation, plasticity, and active change. How you ultimately react to the world depends entirely on which receptor is winning that biochemical tug-of-war at any given moment.
If this tug of war is happening on a single cell, how does that actually translate to me deciding to quit my job? I mean, one cell can't be making that decision alone. We have to zoom out to understand how this microscopic battle affects the entire human brain.
Absolutely. To bridge that gap between the microscopic receptor and the macroscopic brain network, we turn to an absolutely monumental study from 2022.
Ah, the Salvin paper.
Yes,
Wow.
published in Nature Neuroscience by Pier Giorgio Salvin and his colleagues, titled Serotonin Regulation of Behavior via Large-Scale Neuromodulation of Serotonin Receptor Networks.
This paper is a massive leap forward.
It really is.
Yeah.
Because it transitions our understanding from individual receptors to brain-wide networks. They wanted to know how the physical distribution of different serotonin receptors across the entire human brain coordinates complex behaviors.
The methodology they used here is wild. They utilized transcriptomic neuroimaging on data from the Human Connectome project.
Incredibly complex.
To break that down for you listening, they essentially had to cross-reference two massive, totally different maps of the human brain. First, they took resting state functional MRI data from living humans.
Which shows which brain regions are talking to each other.
Right, essentially a map of the city's active traffic grid. Then they overlaid it with postmortem genetic expression maps.
Yes.
This second map shows where the genes that build specific serotonin receptors are actively turned on across the brain.
It's a highly sophisticated approach. They used advanced statistical modeling to find hidden patterns connecting these receptor networks to actual human behavioral scores.
Mm-hmm.
By overlaying those two maps, they discovered what they call serotonin receptor networks, or SRNs.
SRNs.
Yes. They found that different areas of the brain that share similar densities of specific serotonin receptors wire together to form unique functional networks that govern very specific behaviors.
And they discovered two completely independent modes of population variation, right? Two distinct networks that dictate how humans behave. Let's look at the first one, which we can think of as the impulsivity and aggression dimension.
Right. The study found a very specific network characterized by greater functional connectivity of the 5-HT1A, 5-HT1F, and 5-HTTC receptors.
And crucially, this network was explicitly localized in the amygdala and the temporal cortex.
Yes.
So they found this specific admixture of 1A, 1F, and 2C receptors wired together in the amygdala. What did that mean for the actual humans in the study? How did they behave?
It meant a stark difference in behavior. Individuals who showed higher functional connectivity in this specific amygdala temporal network exhibited significantly greater impulsivity.
Impulse.
Yes. The researchers measured this using a behavioral paradigm called delay discounting. In a delay discounting task, a human's offered a choice. You can either have a small amount of money right now or a much larger amount of money a year from now.
It tests your patience and your impulse control.
Exactly.
I feel personally attacked by this study. If you offer me $20 right now versus $50 next year, my amygdala is definitely screaming to take the 20 and go buy lunch.
You wouldn't be alone in that. But humans with strong connectivity in this specific SRN were highly impatient. They consistently discounted the future reward and grabbed the smaller, immediate cash.
But it wasn't just financial impulsivity, was it?
No. These exact same individuals scored much higher on clinical psychiatric assessments for antisocial problems and emotional aggression. So this specific receptor network directly mediates a human's vulnerability to impulsive, aggressive, and antisocial behavior.
But the researchers found a second network, right? One that is completely orthogonal, meaning it's totally independent of that impulsivity network.
Yes. The second mode, which we can call the negative bias and depression dimension. This network involves an entirely different admixture of receptors located in entirely different brain regions.
Which receptors?
This network linked greater functional connectivity of the 5-HT1A and 5-HT4 receptors. And physically, this network was localized in the parietal cortex and the insula.
Specifically a region called the Rolandic operculum.
Yes, exactly.
And the behavioral link for this second network was totally different. While the first network was all about impulsive aggression, this insula network was behaviorally linked to a profound negative bias in reward processing.
They tested this using gambling decision-making tasks.
Right. And the humans with strong connectivity in this parietal insulin network showed significantly slower responsiveness to rewards. It was as if their brains were muted to the concept of winning.
Yes. And aligning perfectly with that, these individuals showed far greater clinical levels of depression, panic, and sadness.
This is a monumental finding because it completely resolves a long-standing paradox in the field of neuroscience.
It really does.
For decades, researchers have observed that serotonin seems to have wildly contradictory effects. Sometimes serotonin dysfunction causes a human to become highly impulsive and aggressive. Other times, serotonin dysfunction causes a human to become deeply depressed, paralyzed by panic, and averse to any risk.
Yes.
How could one single chemical be responsible for both impulsive aggression and aversive depression?
The Salmon paper finally explains it. It is not just about the gross total volume of serotonin floating around in your brain. The behavioral outcome is entirely dependent on the specific admixture of serotonin receptors and exactly where those receptors are located in your brain's geographic architecture.
Right.
If your serotonin signaling is disrupted in the 1A and 2C amygdala network, you become aggressively impulsive. If it is disrupted in the 1A and 4 insulin network, you become profoundly depressed and panic-stricken.
It's like water flowing through a plumbing system. The water, the serotonin, is exactly the same, but if it flows into a pipe that leads to a sprinkler, it waters the lawn. If it flows into a pipe that leads to a boiler, it creates steam. The outcome depends entirely on the hardware it interacts with.
That's a brilliant way to put it.
So we've established the hardware. We know about the 1A passive endurance receptor and the 2A active plasticity receptor. We know about these large-scale receptor networks governing impulsivity and depression. The next logical question for you listening is, what happens if we actively intervene? What happens to a human being's behavior if we artificially flood this incredibly complex system with a massive wave of serotonin in real time?
To answer that, we turn to the absolute cutting edge of human behavioral research. We are going to examine a 2024 paper published in Nature Communications by Michael Colwell and his colleagues.
Direct serotonin release in humans shapes aversive learning and inhibition.
Yes. This paper is incredibly important because of the specific pharmacological intervention they used, which allowed them to isolate the effects of serotonin in a way that previous human studies simply could not achieve.
Historically, studying serotonin in living humans was actually really messy.
Very messy.
You essentially had two flawed options. Option one, use SSRIs like Prozac or Lexapro. But SSRIs don't just dump serotonin into the brain immediately. They block the reuptake transporter, so serotonin slowly pools in the synapse over weeks.
And they cause all sorts of complex downstream effects. They even inadvertently hit dopamine pathways, which totally muddies the behavioral data if you're trying to isolate serotonin.
Right. Option two, you do something called acute tryptophan depletion.
Yes.
You feed a human a terrible-tasting protein drink that lacks tryptophan, the amino acid your body needs to manufacture serotonin. This crashes their serotonin levels.
So again, it's a blunt instrument. And you are only seeing what happens when serotonin is taken away, not what happens when it's increased.
Colwell's team sidestepped all of those historical limitations by utilizing a drug called fenfluramine.
Yes, fenfluramine is what we call a selective serotonin releasing agent, or an SSRA.
SSRI, okay.
It was recently licensed at low doses to treat a specific type of childhood epilepsy called Dravet syndrome. Unlike an SSRI, which just blocks the recycling of serotonin, an SSRA actively forces the neurons to dump their stored serotonin directly into the synapse.
It is a direct, clean, and rapid flood of serotonin.
Exactly. And at these low doses, it does not mess with dopamine levels. So for the first time, researchers could give healthy humans a pill, flood their synapses with serotonin within hours, and then put them through rigorous cognitive tasks to see exactly how their behavior changed.
So they give these participants fenfluramine to flood their brains with serotonin. What was the first thing they noticed when they actually put them to the test? Did their reaction to stress change?
It did, significantly.
Yep.
The first domain they tested was aversive learning and loss sensitivity. They put the participants through a probabilistic instrumental learning task.
To visualize this, imagine you are sitting at a computer and you are shown pairs of abstract symbols. You have to choose one. Sometimes choosing a certain symbol wins you money. Sometimes choosing a symbol causes you to lose money.
But it's probabilistic, meaning the good symbol only wins 70% of the time and the bad symbol loses 70% of the time.
Your brain has to track the outcomes over dozens of trials and figure out the hidden rules, dealing with the frustration of losing even when you made the right choice.
Yes.
I would be so frustrated by that 30% failure rate on the good symbol. My brain would immediately think the rules had changed.
That's the exact frustration the test is designed to elicit. When the researchers analyzed the data using complex computational reinforcement learning models, they found a very specific effect.
What was it?
The humans who had their serotonin artificially elevated by fenfluramine showed a significantly reduced sensitivity to aversive outcomes, meaning the loss trials.
Oh, wow.
When they lost money, their brains essentially registered it as less of a big deal compared to the placebo group.
Yeah.
Their outcome sensitivity parameter for losses dropped significantly.
And this is so crucial because it is the exact opposite of what happens when you take serotonin away. When researchers do those tryptophan depletion studies and crash a human's serotonin, the human becomes hypersensitive to losses.
Every time they lose money on the computer screen, they panic and completely change their strategy.
But here, flooding the brain with serotonin acts as a buffer. It blunts the sting of a negative outcome.
This connects perfectly back to our earlier discussion of passive coping.
Right?
If you are in an environment where bad things are happening, a stressful job, a hostile environment, hyperreacting to every single negative stimulus will exhaust you. By reducing your sensitivity to aversive losses, an acute flood of serotonin helps you endure the situation.
It protects you.
Yes. But the researchers didn't stop there. They also look at a second major domain of serotonin function: behavioral inhibition and impulse control.
This is where they use the effective interference go-no-go task. Let's put you, the listener, in the participant's shoes for a second. You're staring at a screen, and letters are flashing quickly. Your rule is press the space bar every time you see a letter. That is the go command.
Right.
But if the letter is an X, do not press the space bar. That is the no-go command. You have to inhibit your physical impulse to press the button. Now, here's the twist that makes it an effective interference task. While you are trying to do this, distracting images of human faces are flashing in the background.
Some are happy, some are neutral, and some are fearful, terrified faces.
The addition of those emotional faces is critical because it forces the prefrontal cortex to maintain executive control while the amygdala is simultaneously being triggered by the sight of fear.
Exactly. And what the researchers found was remarkable. The humans with elevated serotonin showed significantly enhanced behavioral inhibition.
They were much better at stopping themselves from pressing the button on the X trials.
Yes. When the researchers applied signal detection theory to the data, they found that elevated serotonin caused the participants to adopt a highly cautious, conservative decision-making strategy across the board.
They slowed down, they took their time, their choice impulsivity dropped dramatically.
Right.
But the most incredible part of this finding was how they reacted to the emotional distractors. The fenfluramine group showed their absolute greatest improvement in impulse control, specifically when they were faced with the aversive emotional interference, the fearful faces.
If we look at the network and regional context we discussed earlier with the Salvin study, this makes perfect sense.
How so?
The amygdala is heavily wired with serotonin receptors. When you see a fearful face, your amygdala wants to sound the alarm and trigger an impulsive knee-jerk reaction. But the fenfluramine flooded the brain with serotonin, heavily engaging those 5-HT1A inhibitory receptors in both the amygdala and the prefrontal cortex.
It put the brakes on the panic response.
Exactly. It allowed the prefrontal cortex to maintain executive control, filter out the terrifying emotional distraction, and execute the correct, cautious behavior.
If you're listening to this on your commute right now and thinking about a stressful meeting you have coming up, what the Colwell study suggests is that the serotonin in your system is actively helping you filter out the panic so you can actually drive your car safely or sit in that meeting without running out of the room.
It's literal biological armor against stress.
It really is. And there was actually a third finding in this study, which surprised a lot of people because memory isn't the first thing you associate with serotonin. They used a task called the verbal N-back task to test working memory.
The in-back task is incredibly cognitively demanding.
Yeah.
Letters appear on a screen one at a time. In a one-back condition, you press a button if the letter on the screen matches the letter you saw one turn ago. In a two-back condition, it has to match the letter from two turns ago.
And in a three-back condition, you are trying to hold a constantly updating sequence of letters in your working memory and recall what happened 3 turns prior. It places a massive cognitive load on the prefrontal cortex and the hippocampus.
It does.
I've tried the three-back task before for a different segment, and it physically hurts my brain. You lose the sequence so fast.
It's very difficult.
Mm.
But what Caldwell's team found was that while overall accuracy didn't change massively, the humans with increased serotonin showed significantly faster recall and processing speed, specifically at the highest cognitive load, the three-back condition.
When the brain was working its absolute hardest, the serotonin flood streamlined its processing speed.
Let's synthesize all this behavioral data back to the Carhart-Harris bipartite model of passive coping.
Okay.
What does an acute flood of serotonin do to a human? First, it blunts the sting and the emotional impact of negative losses.
Right.
Second, it makes the human highly cautious, vastly improving their behavioral inhibition and impulse control. Third, it completely filters out the distracting panic caused by aversive emotional stimuli, like fearful faces. And fourth, it sharpens working memory processing speed under high cognitive loads.
When you combine all of those physiological changes, what you have is a brain that has been perfectly prepared, biochemically, to safely endure a highly stressful, dangerous environment. It is the ultimate manifestation of the 5-HT1A passive coping system in action.
It truly is the shock absorbers working at maximum capacity.
But to really bring this full circle, we have to ask how. How does serotonin actually achieve this at the cellular level? We know the behavior, we know the regions, but what are the microscopic mechanics happening inside the cortex to justify this microscopic behavior?
For that, we turn to our final piece of literature, a highly detailed 2013 review paper in Frontiers in Integrative Neuroscience by Pomalata and colleagues, titled Serotonin Modulation of Cortical Neurons and Networks. This paper dives into the cellular mechanics, specifically focusing on the prefrontal cortex. The prefrontal cortex is organized in layers, and the main computing units are the large pyramidal neurons, particularly in layer 5.
Okay.
These pyramidal neurons have long axons that send commands down to lower brain regions, and thick apical dendrites that reach up to the top layers of the cortex to receive incoming information.
So how does serotonin play its receptors like a piano on these specific cells?
Salata details how the 5-HT1A and 5-HT2A receptors are strategically placed to exert maximum control. The inhibitory 5-HT1A receptors are often located on the axon hillock, the very base of the neuron where the electrical action potential is generated.
Think of it as the checkout counter of the neuron.
Yes, when serotonin binds there, it opens potassium channels. Potassium rushes out of the cell, and the cell hyperpolarizes. It strictly downregulates the action potential output. It calms the system down at the source.
And what about the 2A receptors?
Conversely, the excitatory 5-HT2A receptors are largely located out in the apical dendrites, the antenna of the neuron.
Right.
When serotonin binds there, it inhibits those same potassium currents and enhances calcium currents. This ramps up the sensitivity of the dendrites, essentially turning up the volume on incoming synaptic inputs.
So you have 1A clamping down on the output and 2A ramping up the incoming sensitivity. But what about the brakes? Doesn't the brain have local inhibitory cells to stop things from getting out of hand?
It does. The cortex is filled with local inhibitory cells called parvalbumin interneurons. Their entire job is to shut down the pyramidal cells. They are the local bouncers.
But here is the brilliant evolutionary design. Salata highlights that these specific inhibitory interneurons largely lack both 5-HT1A and 5-HT2A receptors.
Exactly. Because these local bouncers are blind to serotonin, serotonin is able to bypass them entirely. It ignores the middleman and exerts a massive, balanced, direct modulation right onto the main cortical networks.
Let's make this highly practical for you listening. Imagine you are exhausted, stressed to the max at that toxic job we talked about earlier. A coworker makes a snide comment, and your immediate emotional impulse is to snap back at them and yell.
But you don't.
You bite your tongue. In that split second, your prefrontal cortex is actively relying on this exact cellular architecture. The serotonin in your brain is completely bypassing the local interneurons, directly binding to the 1A receptors on the axon hillocks of your pyramidal cells, hyperpolarizing them, and hitting the biological brakes to enforce behavioral inhibition.
That momentary decision not to yell is a molecular victory won by your serotonergic network.
It is a profound example of the passive coping mechanism executed through billions of microscopic receptor interactions.
It really is.
Let's distill this deep dive into a summary of what we've covered today. We started by dismantling the myth of the happy chemical. Serotonin is a master regulator of how we survive the world. We learned about the bipartite model of coping from Carhart, Harris, and Nutt.
How the inhibitory 5-HT1A receptor helps us endure the unchangeable, acting as a shock absorber.
While the excitatory 5-HT2A receptor provides the neuroplasticity and the drive to actually change our environment, acting as our steering wheel.
We then map these concepts onto the human brain using Salvin's transcriptomic neuroimaging. We saw how the exact mixture of these receptors across specific brain regions dictates our behavioral vulnerabilities.
An admixture of 1A, 1F, and 2C receptors in the amygdala regulates our impatience, impulsivity, and aggression, while a network of 1A and 4 receptors in the insula regulates our negative biases, vulnerability to depression, and panic.
And finally, through the Colwell study, using the serotonin-releasing agent fenfluramine, we saw that releasing serotonin directly into the human brain literally turns down the volume on negative losses.
It vastly improves our behavioral inhibition, makes us more cautious decision makers, filters out terrifying emotional distractions, and even speeds up our memory processing. Serotonin gears up the human machine to survive the storm.
It is an incredibly elegant, life-saving biological system. But understanding the precision of this system also brings us to a crucial, final, provocative thought regarding the future of our evolutionary biology.
Yes, if our capacity for passive endurance, our ability to just put our heads down and tolerate misery, is mapped to entirely different serotonin receptor networks than our active drive to change the world. What happens as pharmacology advances in the coming decades?
Right, what happens if we get too good at targeting these networks?
If pharmaceutical companies eventually develop a hyper-specific medication that perfectly targets just the 1A endurance network without touching the 2A plasticity network, could we accidentally engineer a population perfectly adapted to tolerate terrible conditions?
From a purely biological standpoint, if you can take a pill that completely eliminates the psychological pain of a hostile environment, the biological drive to escape or alter that environment might disappear entirely.
Can we eliminate the physiological burden of anxiety without accidentally erasing our biological evolutionary drive to adapt and progress?
That is a staggering thought to leave on.
The line between treating biological stress and erasing the evolutionary motivation for progress is razor thin, and it all comes down to which receptors are flipped. I want you, the listener, to mull that over until our next deep dive.
It's definitely something to think about.
The next time you find yourself biting your tongue in a stressful situation, take a second to appreciate the molecular fireworks happening in your prefrontal cortex that are keeping you safe. Thank you so much for joining us on this exploration of the brain. Keep asking questions, keep being curious, and we will see you next time.